Right, yes, I've been asked to talk about exploiting rumen efficiency. I'm not sure it's a thing you can exploit. It's something you can aspire towards and things like that. Hopefully when you achieve it, then you will improve your system. One way of improving dairy cows is to have them all genetically modified with the University of Nottingham logo. They're far more efficient there. Okay, I'd like to acknowledge the AHDB partnership for funding this work. I'd also like to acknowledge my team. I'm not going to read them all out. There's 15 people there. I'm not sure if it was Ray or Jim at the beginning, but they were talking about leverage funding, and only 3 of these people are actually funded by Dairy Cow as it was then, but they've all contributed in various different ways. Okay, so here's our average dairy cow. She yields 30 kilograms of milk and eats 19 kilograms of dry matter a day. If we look at the nitrogen content of her feed, 490 grams, the nitrogen output in her milk, 150 grams a day. So what comes out the back end in the urine and feces is the difference, 340 grams. But 69% of nitrogen that goes into the cow comes out the back end. It doesn't sound very efficient, and it's a source of pollution. On top of that, the cow produces 500 litres of methane a day on average. So it's no wonder that there's this sort of negative image about dairy cows polluting the environment. What we should really be looking at, though, is how wonderful this animal is at converting human inedible food into really top quality food material for humans. So if we look at the human edible portion of the cow's diet, and this is an average cow, so it includes a bit of wheat, a bit of soya, maybe some bran or something that we could eat, then on average across the whole country, the average human edible food intake is 416 grams a day, and the average human edible food output is 589 grams a day of crude protein. That's in milk, obviously. So that makes the cow 141% efficient. So we're getting out 1.5 times as much edible protein as we're putting in. And that's the sort of thing that we should be telling people, not that it produces lots of feces and urine and stuff. It's how great the cow is at converting grass and forage and byproducts into human edible food. And what makes the cow be able to do that is, of course, the rumen. And if we look at the rumen, it's on the left side of the cow. This is a cow from the right side, but you can see all its stomachs. But the rumen's this great big fermentation chamber, the first stomach. Food comes down the esophagus into the rumen, might go up and down again a few times, into the reticulum, the omasum, then the abomasum, which is the same as our true stomach, and then on to the intestines. So within the rumen, it's the microbes that do all the business. So we don't have enzymes to digest cellulose in grass, but there's lots of microbes that do. And the number of microbes in the rumen is truly staggering. There's bacteria, that's 10 billion per milliliter. So each milliliter of rumen fluid contains more bacteria than there are humans on the planet. That's 1 milliliter. Then there's archaea, there's protozoa, and there's fungi. All these microorganisms have different roles, and they all interact with each other and do different things and pass nutrients to each other and things like that. So when food goes into the rumen, these microbes get to work on it, and they produce various byproducts. The main byproduct we're interested in is volatile fatty acids. This is a waste product produced by the microbes, but it's very useful to the cow. The main ones we're interested in are acetate, propionate, and butyrate, and different proportions of them are produced from different feeds. They also produce methane and carbon dioxide, and as we know, methane's a greenhouse gas, so we're very interested in trying to minimize methane production. There's ammonia produced as the microbes break down protein. They produce ammonia, and that goes across the rumen wall if it's in excess. And then going out of the rumen to the omasum, there's undegraded food, and there's the microorganisms themselves. And these microorganisms then feed the cow on high quality energy and protein. So what do we mean by rumen efficiency? We'd like to maximise volatile fatty acid yield. We'd like to maximise microbial protein yield. We'd like to minimise methane and carbon dioxide wastage to the atmosphere. We'd like to minimise ammonia excess, because excess ammonia is converted to urea. Some of it's recycled in the saliva, but some comes out in milk, as you've already heard, and most of it comes out in the urine. So these are the sort of things that we want to do to maximise efficiency. And in terms of undegraded food, that's not really a function of rumen efficiency. We want to balance the undegradable portions of the diet to maximise production relative to what the rumen produces. And just to put it in perspective, the energy supply to ruminants, 70% of it comes from volatile fatty acids, 10% from microbial cells, and only 20% comes from the undegraded food in the diet. We've already heard about protein. When a cow eats protein, some of it's degraded in the rumen, some's undegraded. And the degradable protein needs to be matched with fermentable energy to make microbial crude protein. And it's the balance between these two that maximises efficiency of nitrogen capture and limits the amount that's excreted in the urine. So how does food get into milk? My screen's gone blank.
We've got an issue with the screen. It's just jumping all over the place.
It's a D sub comp 2 problem, I think. Okay, are we back? Right, okay, so looking at the different feed components then, we have protein. Some of that will go straight through the rumen undegraded, but the majority of it will, or depending on the type of protein, but it'll be broken down to ammonia by the microbes and then made into microbial protein. Then they're digested in the intestine to supply amino acids, which go to make milk protein. Starch in the diet is some of it's bypass starch, which is made into glucose in the small intestine. Some of it, or most of it, is converted to propionate in the rumen, and that then goes to glucose in the liver. And as Liam just said, glucose is the major importance in the rumen in the dairy cow, because it's what lactose is synthesized from, and lactose is the main osmotic component of milk. So the more lactose you produce, the more milk you produce. And because of the high demand for glucose, then there is also this pathway of using amino acids as a glucose source as well. If we look at the components in the forage, then we have various different substances. There's pectin, fructosands and sugars. These are the cell contents of the plant, and they will produce propionate, acetate and butyrate. Again, the propionate goes to produce glucose. But we're cycling nicely now. The butyrate and the acetate are used to synthesize butterfat in the mammary gland. And then, if we can ever see it, we have cellulose and hemicellulose, which are the cell wall components, the fiber in the plant. They're broken down to produce acetate, which goes to synthesize butterfat. And the final two bits are lignin, which is undigested in the rumen. It just provides structure for the plant, but it does also provide effective fiber for the rumen. And then finally we have fat, which isn't digested in the rumen, but it may be biohydrogenated to change the fatty acid profile. Okay. Right, I'd now like to advertise this visual hire company in the Midlands. They do an excellent job of putting on presentations. My timer's still not stopped though. Why can't the timer stop? That would be the best thing.
We'll get the timer stopped, don't worry.
It's all right. I have an excuse for running late.
Yeah. Well, while we're waiting, does anyone have any points of clarification or questions for Phil while we're halfway through his presentation? No?
I've got a tricky question.
Yeah, carry on.
Oh, we like tricky questions, Liz.
So for dairy cows, it's 141% in terms of the ability to convert to edible human protein. What's
Yep.
for beef cattle?
Beef cattle. Sorry,
can we?
the human edible conversion efficiency. For dairy cattle, it's 140%. For beef suckler cattle, then it is over 100%, but only slightly over 100. I can't remember the exact number. For all other animals, it's below 100%. In fact, pigs and poultry are the lowest efficiency of all because they have to be fed on high quality proteins and energies. So they eat a lot of wheat and soy and things like that. So ruminants are actually more efficient than non-ruminants in converting human inedible food into human edible food. And the reason suckler beef is so efficient is that they mostly feed them grass and byproducts.
Go back up, which
One before that, maybe?
that's it's a presentation before that. So if you, yeah.
This is a good presentation.
Yeah, it's great. Do you want me to give this one instead? If it's working, then it's better.
Go back up, yeah, that's
Couple
the right
more slides
presentation,
up.
but just back up a few more slides.
Yep.
Yeah, there we go. That's the
37
one.
is the one we want.
Thank you very much.
Patience. Right, okay. So the main project that we've been doing within the AHDB Dairy Partnership is looking at non-invasive indicators of rumen function. Now, I've told you various bits of the rumen that are important, and what we were looking at was what can we actually measure outside the cow to tell us how well it's doing. So feed intake's an obvious thing, and milk yield, but they're pretty routine things. It's got a mind of its own now. I'm not pressing any buttons. But then the more novel technology things, we've been measuring rumination time. It must have some kind of automatic. Rumination time we've been measuring using rumination collars, and that can then sort of tell you how many minutes per day the cow has spent ruminating. It will also tell you it's definitely it may be just the animation's wrong, but if we could stick on this one for now. So this is rumination time in blue. We've also got activity monitors in our cows and milk yield as well. So rumination time dropped this particular day. If we were just looking at that, we'd say the cow may be sick, but actually it was the day the cow was in estrus, so that's why she wasn't spending time ruminating. We also take rumen fluid samples just to see what's going on within the rumen. We do this by stomach tube. We've got a really good system for stomach tubing cows. We measure methane production. I'll show you how we do that later. We measure milk fatty acids and look at the fatty acid profile in milk, which is directly related to rumen function. And we look at cow feces, what comes out the back end. And a really innovative thing we're looking at is cortisol in the feces. Okay, so rumination time then. Rumination, as you know, is chewing the cud, and the objective is for the cow to break down feed particles so they're small enough to digest. But what's even more important is that rumination stimulates saliva flow, and saliva buffers the rumen pH. And the saliva of ruminants maintains pH between 5 and 7, even better between 6 and 7. And without this buffering, the amount of volatile fatty acids produced would make the pH of the rumen between 2 and 4. So you can see the magnitude of the buffering effect of saliva. The flow of saliva is somewhere between 200 and 300 litres per day, and that supplies more than 3 kilograms of bicarbonate per day. So if you think about putting buffers into a feed or something, you might feed 100 or 200 grams a day. The cow would naturally produce 3 kilograms a day. So saliva flow is really important in the cow. Rumination time is variable. We measured on 400 cows. The average was just below 8 hours a day, and it varied from 3 to 11 hours a day. There's a positive effect of forage digestibility. Rumination time has a positive effect on forage digestibility, but not the overall diet digestibility. And that's because there's this relationship between concentrates and forage. Concentrates are more digestible than forage. So rumination time is not a direct indicator of the whole tract digestibility. As I said, it's encouraged by physically effective fibre. People call it the scratch factor. It's not actually scratching, but long fibre particles will stimulate rumination. And cow comfort is very important as well. Cows ruminate a lot more when they're lying down comfortably. The longer they lie down, the more they ruminate and the more they digest their forage. If there's a sharp drop in rumination time, then that usually indicates a sick cow. But as I showed, it may be just a cow in estrus, so don't call the vet immediately. If there's a low overall mean in your herd, then it could mean there's low physical effective fibre. The worst case of low rumination we had was when we started a trial once. We got a really high quality forage, grass silage. Its ME was over 13. It was very short chop length, and we put a high starch concentrate in, and pH just plummeted. They stopped ruminating and all sorts of things. We cured it by 0.5 kilogram of chopped straw. No nutritional value, but the chopped straw stimulated rumination and everything got back to normal again. Okay, so we've got our rumen, we've got it ruminating. Now let's think about the balancing energy and nitrogen in the rumen. And here's a theoretical set of calculations I did where we were looking at the problem with excess of rumen degradable protein. So we designed rations for 40-litre cows based on grass silage alone, a mixture of grass and maize, or just maize on its own. Energy levels the same, metabolizable protein supply the same, crude protein supply dropped a bit, starch went up a lot, but the main thing that we're interested in was the rumen degradable protein to microbial crude protein ratio. So in the grass silage diet, which was balanced for everything, 2 to 1, going down to the maize silage where it was 1 to 1. When we looked at the output in the feces, there was no significant difference between in fecal output. but the urine nitrogen went down as we approached this 1-to-1 ratio, so we were capturing all the nitrogen in the rumen, and overall nitrogen efficiency went up 25%, 30%, 37%. Now that's a theoretical exercise. Kevin has already shown you in the low protein study. There was no difference in dry matter intake and milk yield. With the high energy diet, we got a high starch content. And when we looked at nitrogen efficiency, it went up from 31 to 36 to 37%. So in this high energy, low protein diet, we were matching the fermentable energy and the degradable protein supply, got an improvement in nitrogen efficiency. Going back to volatile fatty acids, they're a good indicator of digestibility of the diet. As I said, we've measured rumen samples in a couple of thousand cows now, and this is a kind of overall analysis of them. And what it shows is the correlations with the overall diet digestibility. And acetate is positively correlated with overall digestibility, and that's because the main thing that comes out the back end is undigested forage. So the more forage that's digested in the rumen, the more acetate produced, and the better the overall digestibility. So that's why there's a positive relationship there. Propionate, though, is negatively correlated, and that's because if you get higher propionate fermentations, you're getting less forage digestion, more coming out the back end. It doesn't mean that you're not getting more energy out of the diet, it just means that it's lower digestibility. I mentioned methane, and we've done a lot of work, and this is what I mean by leverage funding. This was funded by DEFRA under the National Greenhouse Gas Platform, and we've been measuring methane in several thousand cows now. We measure it during milking, so in our robotic milking system, the cow puts her head into a feed bin. We measure the methane production, and once a minute, a cow will belch methane into the tube. And it's regular as clockwork, about once a minute, but it does vary. So this cow's every 57 seconds, this cow's every 50 seconds, and you can see there's a difference in the height as well. We did lots of work and correlated it, the rate at which a cow is producing methane against total daily methane production, and they're highly correlated. So looking again across a lot of data, this is 1,000 cow data. There's a strong relationship between methane production and acetate in the rumen. So this fits with the theory because, well, not theory, with the known biology that when acetate is produced in the rumen, the bugs will produce excess hydrogen. That hydrogen is combined with carbon dioxide by the archaea and produces methane. So digesting forage is what leads to methane production in cows. If you look at propionate, then you get the opposite reaction. So high concentrate diets tend to produce less methane. But these are not high versus low concentrate diets. They are cows all fed on similar diets. The diet effect was taken out. This is just individual cow effects. So this is different rumens produce different amounts of acetate and propionate. Methane is related to milk yield. As you can see on this left one, there's a positive relationship with energy-corrected milk. It's not a perfect relationship, but it's nevertheless positive. And basically what that's telling us is that the more milk a cow gives, the more feed she has to eat, and therefore the more forage she's digesting, and so the more methane she produces. On the right-hand side is a very interesting graph that this, by the way, is our EU project, Rumenomics. We thought we want to reduce methane, and that extra energy that goes into methane will then go into milk. It doesn't. which was a huge surprise to all of us. So as methane goes up, then feed efficiency goes up. So more efficient cows produce more methane. And we've got a big dilemma there. You know, if we take the sort of advice of the environmentalists and the politicians, we want to reduce greenhouse gases, reduce methane, we end up with less efficient cows. And the problem with that is that to produce a given amount of milk to supply the customers, we would have to have more cows, they produce more methane. So looking at it this way round, then cows that produce more methane are more efficient. But if we look at it the other way round and say if we want to produce a certain amount of milk, then we want highly efficient cows because they'll produce less methane overall because they've got less maintenance production. So this presents a really, really interesting scenario. But what this data is, it's ones that are above or below the mean of the population. And what's interesting is there are quite a number of cows in this bottom right-hand side that produce less methane and are more efficient. So we can select cows that are more efficient and produce less methane, but when people tell you to lower methane, remind them this should not be the only goal that we aim for. Right, something else we've been looking at is looking at dung. Laura, you might recognise our microphone carrier. She's spent the past 3 years pointing at bits of dung and looking at it in terms of how good it is, but mainly interested in fecal cortisol. So cortisol is released into the blood when you or a cow gets stressed, and about 10 to 12 hours later, the level in the feces rises. So if you go along and sample a cow 10 to 12 hours after stress, then its cortisol level will be up. And this was confirmed by putting cows through a mild stress event, which was putting them in a crush and blood sampling them. Then 10 to 12 hours later, the fecal cortisol was up. What's interesting is that in the first study, Laura found that when the diet was changed, cortisol went up, suggesting that changing the diet of a cow causes a mild stress. In this case, the energy and protein sources were quite different between one diet and the next. When she repeated it, there was quite a dramatic change in energy and protein sources. But after a few days, then the mean cortisol levels were okay. So it wasn't a dietary effect, it was the stress of changing the diet. But the response was cumulative. The more often the diet was changed, the higher the stress response or the cortisol levels. And if only the silage portion was changed and not the rest of the diet, then there was no effect. So that's kind of interesting. We still don't know exactly whether this is a big stress thing that we need to worry about. We don't think it is, but it could be an indicator of imbalance within the rumen. And we don't know if cortisol yet is an indicator of rumen function. Finally, I want to talk about milk fatty acids. Just briefly, we measure about 100 fatty acids in milk, and there are some that are positively correlated with methane and some negatively correlated. And we've done a similar thing with looking at acetate and propionate as well, but we don't have time for that. But what's interesting and encouraging is that the shorter chain fatty acids, which are made in the mammary gland from acetate, are positively correlated with methane, and the longer ones are negative. And that's shown on this one here, where we had a big population. This is all the short chain fatty acids synthesized in the mammary gland, positively related to methane. And this is milk polyunsaturated fatty acids, and they come mostly from the forage portion. They're higher in a high grass diet, So again, it's positive. So the negative bit was the monounsaturated fat, the oleic acid in the middle. So we can use milk fatty acids as a sort of fingerprinting test to look at rumen fermentation. Finally, I want to share with you some a sort of picture of the future, which is the rumenomics data. This is looking at feed efficiency in 1,000 cows in 4 different countries. And you can see that there's quite a normal distribution of the population. There's a bit of difference between countries, but nothing worth setting up passports for or anything. But the average is somewhere around sort of 1.5 feed efficiency. But look at the range. It goes from 0.6 to 2.4. So that's a huge variation across cows in the efficiency. And what we want to do is to try and move cows up towards this right-hand side, making them more efficient. And cows are certainly individuals, so this is really the sort of count of individual cows. So we can select cows that are more efficient than others. So just to conclude then, my time is up. We can maintain rumination with effective fibre, and that's really important to look at that and keep rumination going. That's what digests the forage for you. Need to balance carbohydrates and rumen degradable protein in order to optimise nitrogen capture in the rumen and maximise microbial protein production. We can adjust starch, fibre and fat supplies to suit whatever milk yield and composition we want for our particular market. So it is possible to switch the type of fermentation as long as we understand what fatty acids are affected in the rumen, and that will then affect milk yield and milk composition. High digestibility forage increases efficiency, so the more digestible your forage, the more efficient your cow overall. But food conversion is actually negatively related to rumen acetate, and starch is used more efficiently in the rumen than forage. Methane is positively related to rumen acetate, so the more forage digested, the more methane produced. We just have to live with it, or we can inhibit it with fat or something like that. But usually when we add fat to the diet, we're reducing the fibre digestion. That's the way high fat works. So to conclude about our overall work package then, we have produced non-invasive indicators of rumen function. They appear to be useful for assessing rumen efficiency, but a message to AHDB dairy, more research is needed to explore them. Thank you.